Hi everyone, my name is Qistina binti Abdul Latif, student ID 2023830032. So today I'm going to present about enzymes. So firstly, enzymes are essential biological catalysts that regulates the speed of chemical reactions in living organisms. They speed up reactions without being consumed or altered in the process, which means they can be reused multiple times. Enzymes are large protein molecules made up of chains of amino acids called polypeptides. Despite their large size, they have an impressive catalytic ability, meaning even small amount of enzyme can accelerate reactions significantly. Enzymes are highly specific in their function, meaning each one is designed to catalyze a particular reaction. However, factors such as pH, temperature, and presence of inhibitors can influence their activity. These factors can affect the enzyme structure and ultimately its ability to perform its catalytic role efficiently.
Hi everyone, I am Kamilah binti Shamsul ini, student ID 2023648174. I will be explaining the application of enzymes in food industry. Let's start with the dairy industry. First is cheese production. The enzyme rennet chymosin plays a crucial role in the cheese making process. It breaks down a milk protein called casein, causing the milk to coagulate into curds and whey. The curds are then processed to create various types of cheese, with differences in texture, flavor, and maturity, depending on specific production techniques. This enzyme has the amazing capability in the production of everything from soft cheeses like mozzarella to hard cheeses like cheddar. Next, we have lactose-free milk. The enzyme lactase addresses a common dietary issue, lactose intolerance. Lactose intolerance affects a significant portion of the global population. So, how does this enzyme work? Lactase will break down lactose, which is the sugar in milk, into two simpler sugars, glucose and galactose. These sugars are much easier to digest. So, this process not only makes milk suitable for those with lactose intolerance, but also retains all the nutritional benefits of dairy products. Moving on to the baking industry. Amylase play an essential role in bread making by breaking down starch in flour into fermentable sugars. These sugars fuel yeast activity, which leads to better fermentation. This process will overall improve the bread volume, give it a softer texture, and create a more uniform crumb structure. Additionally, amylase help extend the shelf life of bread by delaying staining, keeping it fresher for a longer period of time. Finally, we have proteases. Proteases modify the gluten network in dough, making it more elastic and easier to knead. This adjustment ensures consistency in the dough, reduces mechanical effort during processing, and prevents issues like tearing. Proteases also allow bakers to achieve specific textures in baked goods, catering to diverse consumer preferences and product requirements. That's all from me. I will pass the presentation to my groupmate ATHIRAH.
Hi everyone, now I will talk about how enzymes are used in the cosmetic industry. Enzymes are popular because they are natural, gentle, and very effective. They are used in two main areas, which are skincare and hair care. Now let me start with skincare. Enzymes in skincare are great because they work gently without causing irritation. For example, exfoliation. Enzymes like papain, which comes from papaya, help to remove dead skin cells by breaking down keratin proteins. This can make your skin smooth and healthier. Lastly is brightening and anti-aging. Enzyme can reduce dark spots and uneven skin tone by breaking down melanin. It can help you to get more even and young look. Now for hair care. Enzyme improve the health of your scalp and hair texture. First of all is scalp treatment. Enzymes like proteases remove protein buildup on the scalp and clear clogged follicles, which can help hair grow. Lipase enzyme also help to control excess oil. Lastly is hair repair. Enzyme like keratinase repair damaged keratin in hair, making it stronger and healthier. In short, enzymes are a great addition to cosmetics because they are gentle yet effective in improving both skin and hair health.
Hello everyone, my name is Rabiatul binti Harun Ar-Rashid with student number 2023-853-234, and today I'm going to talk about enzyme in food digestion. In our body, there is an enzyme called amylase that are used to facilitate food digestion in our body. So how amylase functions in food digestion? So the first step is amylase binding to starch. Amylase targets specific alpha-1 for glycosidic bonds between glucose units in starch. The starch, which consists of amylose, a straight chain, and amylopectin, a branched chain. Amylose is easier for amylase to act on due to its linear structure, while amylopectin requires extra processing because of its branching. Second is hydrolysis of glycosidic bond, where amylase catalyzes the hydrolysis of alpha-1 for glycosidic bonds, splitting the starch into smaller sugars. The process begins in the mouth with salivary amylase and continues in the small intestine with pancreatic amylase. Hydrolysis produces smaller sugars like maltose and maltodextrose. Next is the formation of intermediate product. The breakdown creates intermediate products such as maltose, maltodextrose, and dextrins, a partially broken starch. Dextrins still contain alpha-1 6-glycosidic bonds from amylopectin, which amylase cannot break on its own. The fourth step is action of debranching enzyme. Debranching enzymes like glycosidase and isomaltase cleave the alpha-1 6-glycosidic bonds in amylopectin. This releases the additional maltose and glucose units, making the starch easier to completely break down. The last step is conversion to glucose. Maltase breaks down maltose into glucose, and other enzymes help break down dextrins into glucose. The resulting glucose is absorbed in the small intestine and transported into the bloodstream for energy.
My name is Mumina Amira binti Salahuddin and my student ID is 2023825692. Today, I will be presenting the topic Catalysis of Biochemical Reaction, focusing on how enzymes play a crucial role in accelerating chemical process in cells. Most enzymes are composed by protein and act as biological catalysts. They speed up reaction by lowering the activation energy EA required for process to occur. By reducing this energy barrier, a great number of reactant molecules can participate in the reaction, resulting in a faster product formation. Importantly, enzymes are highly specific, meaning each enzyme catalyzes a particular type of reaction. For example, lipase catalyzes the hydrolysis of lipid, while sucrase facilitates the breakdown of sucrose in glucose and fructose. This specificity can be explained by a log key mechanism. First and foremost, each enzyme has an active site, a specific region that functions like a lock designed to fit a particular substrate, which acts as the key. When the substrate binds to the active site, a chemical reaction takes place, resulting in the formation of a product. Remarkably, enzymes remain unchanged after the reaction and can be reused multiple times. However, enzymes only function optimally under specific environment conditions such as temperature and pH. When these conditions deviate from optimal range, enzyme activity decreases. This sensitivity, combined with their reusable and efficiency, underscore the essential role of enzyme in sustaining life process. That concludes my presentation on enzyme as catalyst of biochemical reaction. Thank you for your time and attention.